Hello everyone, it's Vesicle here, and today we are going to cover the structure of the animal cell. What we are going to discuss inside this video today is that we are going to discuss the organelles and the function of the organelles inside the animal cells. Before we begin, do follow me on my Facebook and Instagram at Vesicle, and don't forget to like, share and subscribe my YouTube channel. All links are in the description box below. Alright. So what separates the extracellular matrix and the intracellular matrix of the cell is the plasma membrane. So first we are going to draw the plasma membrane. Alright, so we are going to label the plasma membrane. Plasma membrane or cellular membrane, both of it are the same thing. So the main function of the plasma membrane is to regulate the movement in and out of the cells. So basically any substances that wants to enter the cell must go through the plasma membrane first. Alright, if we zoom in this part over here, what we'll see is that we are going to see these tiny circles over here, or these tiny layers over here, known as the phospholipid bilayer. So in the phospholipid bilayer, what we can see is that it has the hydrophilic head and the hydrophobic tail. So the hydrophilic head is this circle over here, whereas the hydrophobic tail is these two lines over here. So the characteristics of the hydrophilic head and the hydrophobic tail is that the hydrophilic head is the water-loving part, meaning that it loves water. That's why it is facing the extracellular fluid and also the intracellular fluid. Whereas the hydrophobic tail is the part which hates water, and that is why it doesn't face any of the fluids. It only faces itself. Alright, see, hydrophobic tail with the hydrophobic tail. So this one is known as the phospholipid bilayer. We would learn more on phospholipid bilayer in the next chapter. Alright. So now let's look into the cell, the animal cell itself. So in order for us to separate, in order for us to separate between the intracellular matrix and the nuclear matrix, what we'll draw is that we are going to draw a nuclear membrane. So a nuclear membrane over here, and inside the nuclear membrane we have chromatins. Alright, so chromatins contains the genetic information of the cell. Basically, chromatins are DNAs. Alright, it looks like this. And in the middle of the nuclear membrane, we have the nucleus. So the main function of the nucleus is to produce ribonucleic acid, known as RNA. So what RNA does is that RNA would decode genetic information from the chromatins and use it for protein synthesis or for production of proteins or enzymes. Next, what we would draw is that we are going to draw the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So this rough endoplasmic reticulum and smooth endoplasmic reticulum are located near the nuclear membrane. Alright. The smooth endoplasmic reticulum and the rough endoplasmic reticulum are an extensive network of folded membranes which form interconnected tubes. So the outer surface of the SER, the smooth endoplasmic reticulum, does not have any ribosomes. So here is the smooth endoplasmic reticulum. Okay. Whereas the outer surface of the rough endoplasmic reticulum consists of ribosomes. Alright, so to differentiate between smooth endoplasmic reticulum and rough endoplasmic reticulum would be the ribosomes. And plus, the ribosomes are scattered everywhere inside the animal cell. So this one is known as the ribosomes. Ribosomes is used for protein synthesis, and this one is the rough endoplasmic reticulum. And this one is the smooth endoplasmic reticulum. So the main function of the smooth endoplasmic reticulum is for lipid synthesis and also the detoxification of drugs. And mainly we found smooth endoplasmic reticulum largely in the liver, because the main function of the liver is to detoxify drugs and for lipid synthesis. Whereas the rough endoplasmic reticulum, the main function of the rough endoplasmic reticulum is to transport synthesized protein from the ribosomes and encapsulate it into a transport vesicle, alright, to travel it to the Golgi apparatus. Now let's draw the transport vesicles. So here are the transport vesicles. Label it. So the transport vesicles would be transported to the Golgi apparatus. This is how the Golgi apparatus looks like. So the main function of the Golgi apparatus is to process, package, and modify proteins that is transported from the rough endoplasmic reticulum. So the Golgi apparatus is like a factory where you would do all those touch-ups and you would add some more features into a basic product. Alright, and this Golgi apparatus will secrete secretory vesicles, which would secrete the modified protein outside of the cell. The next structure that we are going to talk about is that we are going to talk about the mitochondria. So mitochondria looks like this. The next structure that we are going to talk is the mitochondria or mitochondrion. It is an oval or sausage-shaped organelle with a double membrane. The inner membrane is deeply folded, forming a series of projections called cristae. And the main function of the mitochondria is for the site of cellular respiration and also the generation of energy. And energy produced by the mitochondria is in the form of adenosine triphosphate, or known as the ATP. Okay, so next we have the cytoplasms. Alright, a cytoplasm is normal because it is a jelly-like medium where biochemical reaction takes place. So without the cytoplasm, these organelles can't even be here. Alright, so we would label cytoplasms. And apart from that, we also have lysosomes. Alright, so lysosomes are organelles that functions to digest macromolecules or worn out organelles. It is because in the lysosome consists of hydrolytic enzymes which can digest macromolecules and worn out organelles. For example, if we have a damaged smooth endoplasmic reticulum, so the lysosomes would digest it and remove it so that a new smooth endoplasmic reticulum can be built by the cell itself. And the last thing that we are going to discuss inside the animal cell is the centrioles. So The main function of centriole, a centriole looks like this, T-shaped structure. So what does centriole do is that centriole is important in the formation of spindle fiber during mitosis and meiosis. So without the centrioles, the cells cannot divide, and mitosis and meiosis cannot happen. That's about it. That is the structure of the cell. So we'll review it again. In the cell, we have the plasma membrane. The main function of the plasma membrane is to regulate the movement in and out of the cell. And next, we have the nucleus. So the main function of the nucleus is to control the activity of the cell, and it contains the genetic information of the cell. So here we have the nucleolus, which produces ribonucleic acid, which would decode the genetic information from the chromatins and attaches itself to the ribosomes for protein synthesis. And next we have the nuclear membrane. The main function of the nuclear membrane is to separate between the nucleus fluid, the nucleus matrix, and the intracellular fluid. And next we have the chromatins, which are basically DNAs, which contains genetic information. And next we have the smooth endoplasmic reticulum. The main function of the smooth endoplasmic reticulum is for lipid synthesis and detoxification and drugs. Apart from the smooth endoplasmic reticulum, we have the rough endoplasmic reticulum. To differentiate rough endoplasmic reticulum and the smooth endoplasmic reticulum, we can see that in the rough endoplasmic reticulum, ribosomes attaches itself to the RER. And the main function of RER is to transport protein synthesized by the ribosomes. And ribosomes, the main function of the ribosomes is for protein synthesis. And next we have the cytoplasm over here. Cytoplasm is a site where biochemical reaction occurs, and when the proteins are transported out of the rough endoplasmic reticulum, it is encapsulated in the transport vesicle to the Golgi apparatus, where the proteins will further be processed, modified, and packaged into the secretory vesicles. So the secretory vesicles would carry out exocytosis, which is a process where it secrete their contents out of the cells. And next we have the mitochondria. Mitochondria is the site of cellular respiration or generation of energy, and energy is produced in the form of ATP. And we have the lysosomes, which digest macromolecules and worn out organelles. And next, the last and not least, we have the centrioles, which is imperative in the formation of spindle fibers. So that's about it for today. Thank you so much for watching this video. I hope that you can benefit from the structure of the animal cell, basically from this video. Stay tuned for the next video. The next video we will talk about plant cells and everything inside the plant cells. But before we end, do follow me on my Facebook and Instagram at @tutormifzal, and don't forget to like, share, and subscribe my YouTube channel. That's about it for today. Bye.